Thank you very much. Well, as Mike said, my name is Charles Hebert. I work at Southern Research in the Toxicology and Pathology Services Department. I've been there for slightly more than 20 years. The topic that I'm going to talk about today is going to have, I'm sure, some redundancy with what has already been covered in some of the previous presentations. But I thought it would be best to kind of give a basic overview of preclinical, or as Michael said, non-clinical toxicology, because it's not all preclinical. And then end a little bit with talking about some of the specific considerations that you have to take into, that you have to think about when you're selecting animal models for doing your toxicology testing. And please, if anybody has any questions, please feel free to interrupt me. I'll do my best to answer your questions if I can. If I don't have the answers, I will try to get those for you. So just to give you a little bit of an outline about what I'm going to talk about, you know, they say when you give a presentation, tell them what you're going to say, say it, and then tell them what you just said. I'm going to start with a little bit of background on what exactly toxicology is and what is the basis for the need to do preclinical or non-clinical toxicology testing. I will speak very briefly about in vitro toxicology testing because it's a necessary part of the process. Before you can move into animal testing, you have to be able to winnow out some of the candidates that are not going to be appropriate because it's a very expensive process. I'll move on to talk a little bit about in vivo toxicology testing and specifically some of the different types of studies that have to be performed for approval of drugs and how some of those studies depend upon the specifics of the disease indication and the drug itself. And lastly, I'll talk about animal models and some of the things you have to think about when you're making your animal model selections. And I will say that these are general principles. It's not unusual to get to the end of your non-clinical or preclinical toxicology program and get some surprises. So if you ever, and I will give a shout out to Dr. Yamshikov here from Southern Research, because he knows exactly what I'm talking about. If you're ever developing a drug and you get to the point where you're really seriously thinking about filing your IND, you want to be sure you go to the FDA with a very solid testing plan and present that to them and get their buy-in. And sometimes they will surprise you and tell you, no, in fact, you do not have to do that non-human primate study that you thought that you had to do. So it's nice that you get surprises sometimes. Usually not. So background in toxicology. At its most fundamental, the definition of toxicology is that it's the study of the adverse effects of chemical, physical, or biological agents, and I would add radiological agents to that on people, animals, and the environment. That covers a lot of territory. So when you're doing toxicology, you may be in a clinical toxicology setting where you're, for example, doing testing for samples in a hospital, or you may be in the setting that I am, where you're doing safety testing of drugs. You may be working for a pharmaceutical company or for a startup company doing development of drugs. You may be working for a poison control center. There are a lot of different permutations to what you can do in toxicology, but they all end up basically at the same place where you're looking at the study of adverse effects of materials upon living organisms. Some of the key assumptions that you have to keep in mind with toxicology, it's typically not ethical or feasible to do testing on human beings. Ultimately, you do in your preclinical setting or in your clinical setting, but ordinarily, before you get there, you have to do testing models. And a primary assumption of using those models is that other organisms can serve as accurate predictive models of what the effects that you may see in humans might be. Therefore, selection of an appropriate model is critical to accurate prediction of what the effects might be in humans. It doesn't do you any good to select an animal model that has an irrelevant physiological system to the particular drug that you're developing. You can hurt people by doing that, by either approving a drug that may in fact cause toxicity to humans if you tested it inappropriately, or conversely by causing a drug to be killed basically before it can get to the clinic when that drug may in fact have had utility for humans. So it's important to understand the strengths and the weaknesses of any particular model that you choose. So why do we do toxicology testing in the first place? I'm sure this is not news to any of you to cover the basics. In order for any drug to be approved by the FDA, or for any drug to be marketed in the United States, that drug has to be first approved by the FDA. There are other regulatory agencies around the world, in Europe and Japan and so forth, that serve the same function and have the same sorts of regulations. But the FDA is charged with ensuring that drugs that are marketed in the United States are, number one, effective and do what they are supposed to do. Two, they are the conditions of use.
Let me see if I can mute them.
Hello. Hey. Oh, yeah. I was about to run to that.
Wendy, if you can hear me, can you help us mute you?
Uh-oh, hold on a second.
I think that should be it. Okay. And
so in the United States, the drug that you're developing may go to any one of a number of divisions of the FDA, depending upon the intended use of that drug. I've got a couple of examples up here for CDER, the Center for Drug Evaluation Research, is used to, in general, evaluate small molecule drugs. Those are standard aspirin, those sorts of things. But there are also part of the U.S. Food and Drug Administration is the Center for Biologics Evaluation and Research, or CBER, and that's where things like gene therapy vectors and vaccines and not monoclonal antibodies anymore, but they will go to CBER. If you have a veterinary drug that you're developing, it will go to the veterinary division. The intent of the toxicology testing is, as I said, to prove that new drugs are safe, primarily before the first administration to humans. But as Mikey referred to earlier, referred to when she was doing her introduction, toxicology testing does continue beyond the first administration to humans. So part of what the FDA wants to do is ensure that drugs are safe before they allow the drugs to proceed into further clinical trials. Has this class talked about the different types of clinical trials? Yeah. Mm-hmm. So phase 1, phase 2. Toxicology testing continues not only during phase 1, phase 2, and phase 3 clinical trials, but it will also continue to some extent after approval of a drug for marketing, after the drug begins to be sold. And that's critical because, as you know, when you're doing a clinical trial, you can only treat a limited number of patients within that clinical trial. It's extraordinarily expensive to do a clinical trial. And the amount of information that you can get about potential toxicity is strongly dependent upon the statistics and the number of people that you can evaluate that drug in. And it's not uncommon for a drug to be approved for marketing on the basis of phase 3 clinical trials and then get out into wider use where it's not as strictly controlled as it is in a clinical trial setting. Doctors can prescribe that drug for off-label use, and you begin to see people getting types of exposures that were never really tested in the clinical trials. So it's important, even after marketing is approved and the drug starts to be sold, that you continue to monitor the results for potential toxic effects that may not appear when you're looking at smaller numbers of people. The approval of drugs in the United States is driven, or the regulatory basis is the Good Laboratory Practices Act. That is covered in the United States under the Code of Federal Regulations 21 CFR Part 58. It's amazing if you ever look at the actual GLP regulations. The booklet is about this big and about this thick. It's maybe 40 pages long. But there are thousands and thousands and thousands of volumes of interpretation of the GLPs. Other regulatory agencies in other areas of the world, like in Europe, have similar regulations, and in addition, other agencies within the United States, for example, the EPA, have their own sets of GLP regulations. In the case of the EPA for approval of things like pesticides and herbicides. They are similar, but the requirements across the world and across agencies are not identical. So it's important when you're getting into GLP work that you understand the requirements of the agencies that apply to whatever it is that you're testing. And in the end, the definitive preclinical studies, and when I say definitive, I mean the studies that are ultimately the ones that are going to be the basis for the regulatory agency's decision to approve or not approve the drug, those definitive preclinical studies must be GLP compliant. GLP compliant is, in some senses, a state of mind. It can be argued whether some types of testing are or are not GLP compliant. One thing that you do need to remember is that even though you're doing a GLP study, and this is especially true for biologics, that the agency understands that there are some sorts of activities that just do not lend themselves to complete GLP compliance. And if you go into a pre-IND meeting with the FDA with a preclinical testing plan, and you are able to say, these studies are going to be GLP compliant, with the exception that, and explain to them, have a good basis for making those studies non-GLP compliant, the FDA can and often will approve that. So you have to be GLP compliant, but sometimes you don't. So as Mikey was saying, many of the presentations so far have been from people who were speaking from the drug discovery aspect of the drug development process. Drug discovery is very early on in the process. It's a relatively inexpensive part of the drug development process, and in it, you're starting with usually multiple candidates and having to do testing to help you make a decision about which of those candidates are the most appropriate ones for you to advance into further testing. Once you've selected a candidate that you think is appropriate for the target that you want, the next thing you do is you start looking at efficacy in a biological system and safety in biological systems. Before you jump right into your animal testing, you're going to want to start with in vitro testing. So I say that the in vitro toxicology is really the crossover point between the drug discovery process and the drug development process. And it's not, there's no solid line where you say, oh, this was drug discovery, we cross the line, this is drug development. But when you start getting into your in vitro testing, you are starting to get into your drug development. One of the things that in vitro toxicology can do is it can help you provide information on the mechanism of action of your drug. A lot of times when you're developing a drug in the early discovery stages, you see that it seems to work, but you may not know why. And so in vitro toxicology can help you design studies to get a better handle on how your drug is doing what it's doing. I'll give some information, some examples in a little while of that. But it also can provide an indication for you of what you might expect to see once you get into your in vivo testing. And sometimes it allows you to, and this can't be emphasized enough, it allows you to make a decision to terminate that development program before you spend too much more money. I usually include this slide. I don't think I included it in the presentation today, but typically only about 1 in 5,000 drugs, and of course that's a rough number, that ever enter the drug discovery process ever make it to the clinic. And of those, typically maybe 1 in 500 will ever make it all the way through to marketing. So you can see that it's pretty rare, really, for a drug to get all the way through, and it helps you to understand that why drugs are as expensive as they are. Because when you're paying for a drug that's made it all the way through preclinical testing and clinical testing, you're not only supporting the development of that drug, but you're having to support the development of the X thousand drugs that didn't make it. So in vitro methods are widely used for, first of all, screening and ranking chemicals. When you have a bunch of different candidates, you want to have a way of, first of all, saying which ones of these may in fact be efficacious and discard the ones that don't. And then once you have a smaller subset of candidates that do seem to do what you want them to do, you need to rank those in terms of which ones have which are the most efficacious, which ones maybe have some indicators of toxicity, which ones have the best chance of making it all the way through. Sometimes, frankly, that decision is a financial one, as well as a decision based on efficacy or toxicity. In vitro methods can help you study cell, tissue, or target-specific effects, and the results of your in vitro testing help you to refine the subsequent study designs. Good things about in vitro testing: they're less expensive to run than in vivo studies. They typically take a shorter time to run than in vivo studies, plus, and this is no small consideration, most of them don't bite. The downside is that your in vitro results are necessarily less predictive of what the toxicity in an intact organism might be than what you would get if you start testing in animals.
So is it possible that sometimes you should in vitro you got very toxic, but in vivo it's less toxic, or even?
Absolutely.
Okay. So that means you need to do both before exclude the candidate.
Not necessarily. A lot of times you can see toxicity in an in vitro system that makes it very clear that no matter what you do with that that drug is going to be toxic in vivo. A good example would be a cytotoxic drug. If it kills cells in vitro, there's a very good chance that it's going to kill cells in vivo as well. I think the most important thing is that when you're doing your in vitro testing, you understand the relevance of the system that you're choosing to do your testing to whatever disease, whatever organ system it is that you're going to be targeting in vivo. You may be able to do some in vitro testing and observe significant toxicity and say, but perhaps that toxicity is due to some mechanism that is not going to be in play in vivo. There may be a detoxification mechanism in an animal that's not present in your in vitro system. But just in general, the simpler the system that you use to test your drug, the less predictive the effects of that drug, of that test are going to be for your in vivo testing. As I said at the beginning, your toxicology testing is all predicated on the assumption that what you're doing is going to be predictive of what's going to happen in a human. And you have to understand the limitations of whatever systems you're choosing to use to do your testing. So some types of in vitro toxicology testing would be cytotoxicity testing. If I put this drug into solution and mix it with Hep2G cells, does it tear those cells apart? Does it punch holes in the cell membrane? Do the cells thrive? There are a number of different types of tests that you can use to look at cytotoxicity, and I'm not going to go into those, although those are slides that I took out of this presentation. Another type of test that you have to do is protein binding. If your drug is going to be administered in any way systemically, and that could be whether it's going to be administered intravenously, is it going to be administered intraperitoneally, is it going to be administered orally and then absorbed into the circulation, you have to understand the effect of protein binding. I remember when I first heard about this, it seemed to me like it couldn't be that relevant until it's explained to me that, say you have a drug that binds to albumin, and the drug in the circulation is 99% bound, and only 1% of that drug is free. So it's only the free drug that's actually going to be able to have any physiological effect. As long as the rest is bound to albumin, it's not going to be able to do much of anything. But what if you then administer a second drug that also binds to albumin and displaces some of the original drug from the albumin? And say it displaces 1% of the drug that was originally bound to albumin. Well, that doesn't sound like very much. It's only displacing 1%. Bear in mind that your original systemic concentration of effective drug was 1%. You have now displaced another 1%, and you have doubled your effective concentration in the blood. So you really do have to understand whether your drug is going to be bound to protein in the circulation, what proteins it's going to be bound to, and what else might be competing for those binding sites. Cytochrome P450 inhibition or induction, I don't know whether y'all have been talked to about cytochrome P450. This is critical. A lot of drugs will induce the production of liver enzymes that then turn around and metabolize that drug. So a dose of a drug that may be effective after the first dose may cause enough enzyme induction and enough metabolism of your drug that you give the same dose again, most of it's metabolized and becomes inactive. So you have to be aware of the effects of cytochrome P450 inhibition or induction. And that's, there's no time to go into that in a lot of detail, but bear in mind that metabolism of a drug can have 3 different effects, basically. It can inactivate that drug. If you're starting with an active drug, it can inactivate that drug. It can basically have little effect on the activity of that drug at all. Or it may, in fact, metabolize that drug to a more toxic metabolite. And it's not a small consideration. Anybody who's taken a lot of acetaminophen knows that. Acetaminophen is a classic example of a drug that's metabolized in the liver, and this is maybe veering off a little bit into the toxicology lesson, but it's metabolized in the liver ordinarily by a relatively low capacity enzyme system that metabolizes the drugs to inactive metabolites. So as long as you're taking low doses of acetaminophen, that low capacity pathway is able to handle those doses, and acetaminophen is not going to be toxic. However, if you take too large doses of acetaminophen, you overwhelm the capacity of that primary pathway, and there is a secondary pathway of metabolism that goes through the cytochrome P450 system that produces a highly toxic metabolite that ultimately damages the liver cell membrane. So understanding the effects of your drug on drug metabolizing enzymes in the liver and other parts of the body is a critical part of in vitro toxicology testing. Looking at membrane permeability. Acetaminophen is another good example. Basically what it does is it punches holes in the liver cell membranes. You want to look at the stability of the drug. Once you administer it, how quickly does it disappear? And the metabolic stability leads to, or is intertwined with, interspecies comparisons. And this is relevant to selection of the most appropriate animal model. One of the ways that you look at metabolic stability of a drug is to use isolated microsomes from various species. Are you familiar with microsomes? Okay, so you know microsomes are small subcellular bits that contain drug metabolizing enzymes, and you can use those in a test tube to evaluate the rate of metabolism of your drug by those particular enzymes, and to look at what the specific metabolites are. When you're looking at metabolic stability, you want to look at how quickly, well, number one, does this particular enzyme metabolize this drug? Number two, how quickly does this enzyme metabolize the drug? Number three, what are the metabolites? And then by extension, you say, are the metabolic effects for my animal model that I'm choosing the same as the metabolic effects that I can expect to see in a human being? Not all mammalian species have the same distribution of drug metabolizing enzymes. Most of them are very similar, but they're not identical. And I can give an example. We did some studies a few years ago for the National Cancer Institute looking at a drug for prevention of cancer. And one of the first things we did was we looked at metabolic stability, and we did a comparison of metabolism in mice and rats and dogs and non-human primates and human microsomes. And the sort of default for selection of species when you're doing toxicology testing of small molecules is to use rats or mice as your rodent species and then dogs as your non-rodent species. Well, we found when we did the interspecies comparison that the metabolites that came out of the the dog microsomes were completely different from the metabolites that came out from the human microsomes. And that told us that use of the dog would not be appropriate for the toxicology testing of that particular drug. We had to use non-human primates, even though that's not what you prefer to do. But had we not done that in vitro testing in that interspecies comparison, we could have tested that drug in dogs and said it looks perfectly safe, and had that drug get out into the clinic and started to see adverse effects that we should have been able to pick up. This is just an extension of some of the things I've said. Cytotoxicity basically is toxicity to cells. Basically, does your drug kill cells? You can use a lot of different types of cells for cytotoxicity testing, and they can be used to assess a number of different things, such as viability, structural effects, and/or function. For example, see if I can use this mouse here. Let's see. One of the most important things that you want to look at is, does my drug cause the cells to increase or decrease their proliferative rate? If your drug does cause hyperproliferation, there's a decent chance that it may ultimately be a carcinogen, or at least a mutagen, and that's important to know for farther on down the road when you. If you're developing a drug that's going to be used for long-term administration, like a drug for treatment of diabetes that somebody's going to take for every day for the rest of their life, you need to know what the long-term effects of that drug are going to be. And if a drug increases cell proliferation over the long term, that presents a high risk for carcinogenicity. Some of the tests that are used currently for in vitro toxicology are developed to replace older in vivo tests. And a good example of that is the dermal corrosion test, the skin irritation test, the Dray's eye irritancy tests. All these used to be done in rabbits. Toxicology by its very nature is looking for adverse effects. But anybody who uses animals knows that you want to use those animals responsibly. You want to use those animals as humanely as possible. And if there's a way of evaluating the possibility of skin irritation using an in vitro model rather than using animals, it's by far preferable to do the in vitro. And there are a lot of in vitro tests that are available now in kit form. People have come up with some really ingenious ways of combining different cell types to do these tests. I have just an example of one here. It's the Epiderm kit. It's composed of normal human epidermal cells that are cultured on a polycarbonate membrane that's permeable, and that polycarbonate membrane sits atop the liquid growth medium so that the growth medium can come up through the bottom, and those cells get their nutrition from underneath, just like they do in normal skin. Whereas in a cell culture, of course, your cells are typically covered over and they're getting the nutrition from the top. The neat thing about the Epiderm kit is that the keratinocytes undergo the same sort of differentiation, stratification and differentiation of different types of cells that you would see in a normal epidermis. So if you take a cross section of the cells in the Epiderm kit, you would see the highly proliferative cells at the base on the polycarbonate membrane. You would see cells farther up in the layers that are starting to flatten out to produce keratin, and ultimately you would see the cells at the top that are just basically the dead cells that are starting to flake off. They're metabolically active, they're mitotically active, they're organized into the differentiated layers. It provides a really good indication to you of what the potential skin toxicity might be. Okay, it's not altogether predictive, but it really it's a nice tool. I've already mentioned in vitro toxicology. One of the things that's important is to assess the ability of the chemical to produce metabolism of specific substrates, including the drug itself. It provides information about the metabolic pathways, whether cytochrome 50 is or is not involved. Provides information on whether your metabolism of your drug is going to produce something more toxic or reactive or possibly less toxic. And again, I've mentioned already the interspecies comparison. Better move along. So moving on into in vivo toxicology. You have several goals for your testing in animals. One of the primary goals is to establish what your starting dose in a clinic is going to be. That can have huge implications on the amount of money that you spend on your clinical trials. One of the typical models for doing clinical trials is to start at a demonstrated safe dose in humans. And if you treat a few humans with that and you see that there's no evident toxicity, you can then get permission from the FDA to move up to a higher dose and continue that escalation until you do reach dose-limiting concentrations, or dose-limiting effects. If you choose your starting dose way down here, and you have to go through 7 iterations to get to the point where you know what your dose-limiting toxicity is, you can spend a lot of money on what's essentially wasted testing. Whereas if you had planned your preclinical testing properly and selected your dose better, you might have been able to start here and save yourself several $1 million just by planning well. So one of the main goals of your in vivo toxicology testing is to establish the safe starting dose for clinical studies. Of course, the flip side of that is you don't want to choose a dose to start with that's too high, because then you're going to start to see toxicity in humans, and that will kill your drug fast. One of the other things that you want to get out of your preclinical toxicology testing is information on what is the most appropriate drug treatment regimen that would produce the least toxicity. We tend to think of drugs as something that you take every day for a set amount of time. You take an antibiotic once daily for 50 days. But that's not necessarily always the proper model for dosing. And so part of your preclinical testing is to see, do I get the same efficacy but less toxicity if I maybe dose the animals with half the dose level twice a day? Or maybe I could raise the dose a little bit and just give it every other day. There are a lot of considerations that go into that in terms of accumulation of toxicity and persistence of the drug. But one of the things you want to learn from your preclinical tox is what is the most appropriate drug treatment regimen. And this is, I'll just mention that this is a pet peeve of mine. It's not a drug treatment regime. It's a drug treatment regimen. A regime is South Korea, North Korea. One thing you want to get out of your in vivo toxicology testing is you want to get an idea of what your target organs may be. That's so that when you do your testing in humans, you can more intelligently plan what sort of monitoring you're going to do in your clinical trials. If you know that there's a potential for liver toxicity, then you know to look for that when you do your clinical trials. And also, you want to get an idea of reversibility of toxicity. We tend to think sometimes that if a drug is toxic, then that's it, redline that one, get rid of it, start with something else. But that's not always the case. There are acceptable levels of toxicity. And the acceptable level of toxicity may vary depending on your patient population and the disease indication. But it's possible for you to get toxicity from a drug that's acceptable if you know that once we stop taking that drug, that toxicity is going to reverse itself. The benefit from the efficacy against the disease may outweigh the toxicity that you see. And again, your in vivo testing should provide some insight into biomarkers, that just things for you to look for in your clinical monitoring that may be early indicators of trouble, so that you don't have to wait till your patient is throwing up or complaining of abdominal pain or something. You may be able to pick up on biomarkers, enzyme levels in the blood that can give you some warning that something's going on. So the types of testing that are required depend entirely upon the drug that you're developing, the disease indication, and the patient population. Typically, you'll start out with single dose, and it's often called acute toxicology testing. Those are used interchangeably, although they probably shouldn't be. Where you're essentially going in blind. You have done your in vitro testing. You selected what you think is an appropriate animal model, but you've never put this into a living organism before, and you really don't know what's going to happen. So ordinarily what's going to happen, well, I think I give you details on the next slides, but with the single dose test, you're going to choose a very broad range of doses so that you can pretty much assure yourself that you're going to get at least 1 or 2 levels that are non-toxic that you can continue with. You're also going to use relatively few animals because you're not looking for a lot of statistical power in these tests. You're basically looking to see, do I see any overt toxicity? Once you have the results from your single dose testing, you can move on to your repeat dose, what I call pivotal testing. And I'll talk a little bit more about these, so I'll skip over that. You also are going to want to do your toxicokinetic and your pharmacokinetic evaluations. That's looking at things like distribution within the body and disposition. I should have put up here what's called ADMET, drug metabolism, absorption, distribution, metabolism, excretion. Pharmacokinetics look at how quickly, basically, the drug is eliminated from the blood. You can do pharmacokinetics on tissues as well, but usually it's in the blood. I will point out that, and so I probably am bouncing around a little bit here, but toxicokinetics and pharmacokinetics are not the same thing. People often use them interchangeably, but and they don't have really firm, solid definitions. But when you're doing a pharmacokinetic study, basically what you're doing is you're collecting serial blood samples over time, looking at the concentration of your drug in each of those samples and plotting out at what time do you have the maximum concentration, what is that maximum concentration, and how quickly does that drug disappear from the circulation. When you're doing a toxic, and those studies are typically done at non-toxic drug level. Because what you don't want to do is you don't want to do your pharmacokinetic study at a dose that causes overt toxicity that could then affect the kinetics of the drug level. Toxicokinetics, on the other hand, typically is done within the context of a toxicology study. There usually are not as many samples drawn. Sometimes you only draw one sample to get a good idea of the fact that you've actually exposed the animals and what the maximum concentration might be. But you're typically not doing a complete evaluation of the area under the curve, the half-life, those sorts sorts of things, like you do in a PK study. Most drugs are going to have to be tested eventually in some specialized toxicity studies known as safety pharmacology studies. Those are studies that are designed to look at toxicity or adverse effects in specific physiological systems. For example, cardiovascular toxicity goes a step beyond what you look for in a normal toxicology test. Just as an example, in a large animal test, physical toxicology study, you will probably have to look at blood pressure and ECGs. But you're not going to be looking for prolongation of your QT interval or other changes like that, per channel tests. So if you do your toxicology testing and there's any indication that there are cardiovascular effects, you're going to have to turn around and do a much more in-depth evaluation, and that's in a safety pharmacology study. Respiratory system and central nervous system are two of the others that are frequently tested in safety pharmacology testing. Sometimes you can combine the safety pharm with your toxicology testing. I would say that most often you really need to do those as standalone studies. Local tolerance testing is something that you do for a drug that's going to be applied to the skin or into the skin or intramuscularly. So if it's something you're going to be injecting IV or the vaccine you're going to be giving intramuscularly, local tolerance is basically a more focused evaluation of the dosing site to see whether there's any adverse effect there. And usually, if it's a vaccine, there is. Usually you see some sort of inflammation there. It's not necessarily a killer, but you need to know that it's a possibility for your clinical trials. And another type of testing that's typically required is genotox testing. Some of this is done in vivo, some of it's done in vitro, but it's basically looking at the ability of your drug to damage DNA. So the number of studies that you do and the type of studies that you do depend on your therapeutic indication. One thing to bear in mind, drugs that are being developed for life-threatening illnesses typically require less testing and it's a higher tolerance for toxicity. A good example is drugs that are being developed for cancer. If your choice is not take the drug and die, or take the drug and have herbal sores in your mouth and lose all your hair, that may be acceptable toxicity if it's going to save your life. But on the other hand, if you're developing a drug that's an analgesic for children, sores in your mouth and losing all your hair is an unacceptable level of toxicity. So the level of tox tolerance varies depending on the indication. Usually, preclinical studies have to be done in 2 animal species, 1 rodent and 1 non-rodent. As I said, the default is usually the rat and the dog. There are practical reasons sometimes for using one over the other, but it's best if you make your decision based on biology rather than dollar signs or convenience for your chemical staff. One exception is that a lot of biologics require only 1 species. And the reason for that, or one reason for that, is because by their very nature, the way a lot of biologics work, their mechanism of action, a lot of species are not relevant. And so it would be useless to require testing in 2 species if there's only 1 species that you can find that's particularly relevant to the testing. Sometimes other species are used. I give examples here for rabbits, ferrets, hamsters, mini pigs. They may be used for special types of testing. Cotton rats are another good example that you don't ever want to use if you don't have to. And ferrets, for example, are good flu model. You usually start with single dose testing. I've already talked about that. You're looking for your most appropriate dose range for your subsequent tests. And so you start with a broad dose range. You use a minimal number of animals. You do a minimal amount of evaluation. Usually just you look at body weights, mortality, of course. Does this drug cause any clinical signs, overt signs of toxicity? You're typically, in your acute studies, not going to do clinical pathology testing to look at hematology, clinical chemistry, coagulation. You're not going to do ECG testing, those sorts of things. And usually the single dose studies do not have to be GLP compliant, or the range finding studies do not have to be GLP compliant. Let me amend that. Because if your drug is intended for single administration use, then you are going to have to do a single dose GLP compliant study eventually. Once you have an idea of what your toxic and safe range of doses is, you move on to your repeat dose toxicity studies if they're required. What I'm going to talk about here will apply also to single dose studies if you have a single dose drug. These are more complete studies, and the preclinical studies should mimic as closely as possible the way you plan to use this in the drug, which means you should use the same route if possible. It's not always possible, but if possible, do so. Same duration of testing and the same schedule of testing. As I say here, the requirements vary between the different regulatory agencies. Typically, for small molecules, we will have to do 2 species, and biologics just 1. Your repeat dose toxicity testing is going to have a much more extensive evaluation. You're going to look at body weights, clinical signs of toxicity, typically food consumption, hematology, clinical chemistry, coagulation effects under clinical pathology, and one of the most critical things to look at is your histopathology. That's where you take your tissues, you prepare your slides, you have a veterinary pathologist look at the tissues and see whether there are any microscopic changes. That's, I would say, typically the most important piece of information for a preclinical study. Large animals will undergo more extensive evaluations. You can do ECGs on a dog fairly easily. You can't do ECGs very readily on a mouse. So the agency understands that, they're not required. You should select your dose range so that at least one dose produces dose-limiting toxicity, and at least one dose should be non-toxic. The reason is that the agency wants to know, when they see your dose range, that you have gone as high as you can possibly go. And if you have not demonstrated toxicity of some kind at your highest dose, the agency may make you go back and redo your test again, because they will consider your test inadequate because you haven't necessarily gotten a good idea of what the toxic effects of this drug may be. Now, it may be that you've tested the drug at the highest feasible dose. There may be limits on solubility of the drug, limits on the volume that you can administer to the animals. And if you can demonstrate to the agency that you have gone as high as you can go and you still haven't seen toxicity, then they may let you proceed into the clinic, but I can guarantee you they'll let you do so at low doses. I've already talked about safety farm, chronic toxicity, carcinogenicity testing, and drugs intended for long-term administration to look at whether they have the potential to produce cancer. They usually use a lot of animals so that you have good statistics and you have a good chance of picking up a low incidence effect. And then finally, reproductive toxicity testing and teratogenicity. We do a lot of reproductive tox testing at Southern Research. It's a very specialized field of toxicology, very complicated. I'm fortunate that I get to stand by and sort of administer those without having to be the study director on those. Biologics require some of the same tests, but each one has its own set of unique requirements. A lot of times they require different animal models. For example, if you're testing an adenovirus vector, adenoviruses are very commonly used for gene therapy vectors and for vaccines. Your animal models of choice are either the hamster or the cotton rat, typically. The reason for that primarily is that those two models are sensitive to infection, or they're susceptible to infection by human adenovirus. And so you want to know, especially if you're using a vector, if I administer this drug to the animal, is there any possibility of recombination so that I get a more infectious agent or a more toxic agent than what I actually administered? If you're giving it to an animal that it's not susceptible to infection, you won't pick that up. But if you use a hamster or a cotton rat, you can. And I would say again, cotton rats, if you get a chance to not use them, they are vicious animals. I've already hit my end, but I would like to just blitz through a couple of these slides. When you're selecting your animal models, these are your assumptions. You assume that they can serve as accurate predictive models. Selection of an appropriate model is key to accurate prediction in humans. You have to understand the strengths and weaknesses of your model, and drugs that show safety and efficacy in preclinical animal models may have very different pharmacological effects in humans. The FDA more and more is requiring sponsors to provide data to justify their selection of animal models, rather than just using defaults. Some of the reasons that a model may be inappropriate, there may not be an appropriate drug target in your animal model. If you're looking for a drug whose mechanism of action is binding to a specific cell surface receptor, and that cell surface receptor is not present in mice, then it's useless to look at mice as your animal model. There may be presence of an irrelevant target, differences in metabolic fate, like what I described a while ago for the drug for the NCI, or differences in susceptibility and infection by specific pathogens, like I just described for adenoviruses. An example of lack of appropriate drug target. Don't test drugs for prostate cancer in women. Really. Presence of an irrelevant target. This one was a biggie 30 years ago. Studies were done looking at unleaded gasoline toxicity and in rats, male rats, there was a very specific type of nephropathy observed, the serious nephropathy. And a lot of investigation went into it, but the pathology consisted of accumulation of droplets, hyaline droplets, which are basically clear inclusions inside of the cells, containing a protein called alpha-2 microglobulin in the proximal tubules. That led to cell death and complete denudation of the lining of the proximal tubules and subsequent inability to reabsorb materials from the urine. However, the same thing wasn't seen in female rats, it wasn't seen in mice, and it wasn't seen in non-human primates. And it turns out that alpha-2 microglobulin is a male rat specific protein. It's not found in humans. So that suggests that the results of that test in rats are not relevant to humans because it's an irrelevant target. I think I'll stop here. I just had a couple more examples. I already talked about adenovirus vectors. Bottom line, pick the right animal model. You're wasting time, money. It's unethical and potentially dangerous to humans to use the wrong animal model.
All right. Thank you so much. Any quick questions? If not, thanks again for coming. I've seen many faces, many weeks, so I appreciate all of you that came to most of the lecture, and I hope you learned a lot from it. And I'm always open to feedback, so let me know what you thought, whether good or bad, or, what you thought of it. With that, enjoy the sunshine.
I kind of blitzed through this. If anybody wants to stay and ask anything specific, I'll be happy to do that.
Thank you. Wonderful, thanks so much. It's challenging. You want to share too much information, but yeah, go ahead. Let me close this out. Would you be willing to share your slides so we can put them on our website? Great.
Everybody sign in?
Yeah.
Where are these going to be posted on? I wasn't able to find.
Because I haven't gotten around to
Okay.
that.
Okay.
That's on the ADA website, I only posted last year's, but I've been so busy with other things, so I figured after the series is complete,